Okay. Yeah. So today we are here with Yuki Ten. She is a postdoctoral researcher in Dr. Gary Nolan's lab at Stanford University and holds a BS in cell and molecular biology from the Chinese University of Hong Kong and a PhD in computational biology from Johns Hopkins University. She specializes in crafting computational tools that integrate and transfer knowledge across various single-cell data modalities, both in 2 and 3 dimensions. Today, she's going to talk about how spatially resolved multiplexed imaging data analysis unveils insights into intricate, spatially organized niches and tissue architectures. Existing hurdles in this field encompass software fragmentation, processing bottlenecks for large images, and a dearth of computational workflows for comparing experimental setups. And so to tackle these challenges, they introduce Spacey, a Python-based end-to-end workflow that adeptly manages extensive imaging data analysis and furnishes a thorough computational pipeline for intercondition comparison. And with all that being said, I hand it over to Dr. Tan.
Thank you so much, Hector, for the introduction. And thank you so much, Ahmed, and other in organizing committee for inviting me. It's truly an honor. So I will present Spacey, the the Python workflow for analyzing multiplex imaging and processing. But what I would sort of frame this talk is in the context of sort of general the larger big picture question that I've always been interested in. So to start with, maybe a sort of like the 3-dimension or 3 projects that I will be sharing. So the first one is really getting at we are in the field of the era of single cell. So the first question that I was always interested in is what defines a cell type, the cellular identity. When people started, it's mostly starting from single-cell RNA-seq, but as we are able to measure more modality and more molecules with high throughput, we are able to define it even better. With the rise of spatial technology, that we now can see the cellular identity and how they interact in their native context, in particular, pertaining to their spatial context. And then that's the second chapter that I'm interested in, which is the cellular neighborhood, the cellular community, and how they are organizing into the functional niche and supporting tissue functions and organ functions. Last but not least, this is a very ongoing effort, and the direction for the next few years for me is that how do we see these cellular interaction and how these cellular differences in the 3D space. So to start with, this is going back a little bit on time, touching a little bit of some of my PhD work. So back in the days, which is only a decade ago, where people developed this technology where you can extract a tissue, dissociate these individual cells from the tissue, and then sequence them, either using, for the RNA contact or for their DNA content, and then use dimensional reduction method to project all these features that has been detected into, let's say, a 2-dimensional space using tissue, t-SNE or UMAP. And the question is that once you dissociate it from this tissue, you don't really know the identity of any one of these dots here. And given the quantity of these single cell, it's also unlikely that we can do the traditional method of looking at each cells and trying to decide what they are. So during my PhD, one of the methods that I developed is called single cell net. We would basically take a single cell RNA-seq training data provided with annotation and train a single cell net, which is a random forest-based machine learning classifier. So if you see the general framework here is that you have the gene expression matrix, and then we do the pair transformation. This is helping to compare single-cell RNA-seq data across platform and across species. And then we select the features that are most differentially expressed, and then we train a random forest classifier. And then with the query data that you want to annotate, you'll be able to get a score for classification and visualize it in various manner. So this is one of the way in which that one can annotate single-cell RNA-seq based on their gene expression data. Since then, for example, based on this wonderful review published in 2019, there are a slew of single-cell RNA-seq annotation methods, whether that's for RNA, for ATAC, for many different methods. Machine learning has really been helping to identify the identity of the single cell. Well, Not so long ago, with the development of single-cell RNA-seq, the technology has also expanded to capture the single cell in the spatial context. So this is another review from Lewis from Nature Method, 2021. So in here, the x-axis is year, and y-axis is the number of cell sequence, and each of this bar here representing a technology that is able to capture their spatial omics, and whether that be DNA, RNA, protein, and cell. And in the Nolan lab we're part of, we contributing to a few of the technology, namely MIBI. It's a and codecs, right? So these are the technology that we use a lot of in the lab, which is sort of also motivating why the package that I'll be introducing today. So why spatial context? What does it help in terms of our understanding of the cell type identity or function? The answer is a solid yes. So we also I helped develop this deep learning method or graph-based computational method called Stella that actually annotate single cell in the spatial data by leveraging the spatial information as well. Briefly speaking, we have a reference data set, which is a spatial data. We tested it on Merfish as well as Codex. You constructed a spatial cell graph, and each one of this node here is a cell with the features both for the protein expression. And you train a graph neural network and a Stella network. And then if you have an unannotated spatial data set, you also constructed a graph based on the distance, and you push it through the previously trained Stella graph neural net model, and then we're able to annotate cell type that's in the training data, but also identifies clusters of cells that are novel and not seen by the data set before. So the difference that now we are seeing is that we are starting to have a lot of machine learning and deep learning method that incorporates the spatial information into the annotation. So does it perform better by having that spatial information? The answer is also yes. So as you see here, we did in the paper, we have a comparison between Stellar and other machine learning algorithm that does single cell annotation. And the y-axis here is the annotation accuracy. So you can see that compare Stella to the next best performing annotation method, which is XGBoost. It's 61% increase in terms of accuracy. So that's definitely signaling that there are information captured in the spatial context that's determining or facilitating the determination of cell type identity. And more convincingly, so when we use both spatial and molecular data for this the cell type annotation, it's an 11% increase in terms of accuracy for annotation compared to just using molecular information alone. So this is definitely saying that with the capturing the cells in their spatial context is a one step further for us to understanding how cell functions and how organized in the tissue context, and help us to better define specific biological questions. So now we know that cell type identity is defined in its spatial context. In addition to understand how that interact to the cell type, the cell itself, how does it interact with its neighbor? How do they form a niche or cellular neighborhood that at large drives the function of the tissue is the next direction that I wanted to introduce. So in this particular context, I will introduce Spacey here, which is the tool that help us to extract information from the spatial context and providing from end to end, starting from the tissue extraction, cell segmentation, all the way to advanced spatial analysis. And this tool in various form has been applied to many different tissues, starting with the brain data, which Yan, we have collaborated and presented a few seminars ago, and then melanoma, it's ongoing work. And then we also apply to human intestine and many other different tissue samples and have obtained quite smooth success across. So let me just introduce a little bit. So what is the bigger context? So many people many people are probably familiar with Codex is. Codex is an iterative multiplex proteomic technology. So you can typically stain it with 50 to 120 antibodies. You cross-link these antibody, which is conjugated with DNA oligos to the cells. And then with these oligos, you have a complementary oligos with different fluorophore attached to it. And you image once, and you denature it, and then you repeat this cycle of using different oligos and imaging different antibody, and then you obtain a multiplex imaging such as this one. So this is an example images that I'll be using throughout explaining how the package works. So these two are tonsil tissues, and this is a regular tonsil, and this is a tonsillitis. So one of the challenges that I encounter, and many also felt that way, is that it's very difficult to analyze image-based spatial omics, because I think traditionally a lot of single-cell RNA-seq technology or single-cell technology can be seamlessly transferred to spatial omics technology that are sequence-based. But there are slightly different pre-processing that's involved, and also transformation is different when it comes to image-based. So how can we extract information from these multiplex images into a baseline understanding of our technology, as well as a figure-ready format is what we're trying to do. So we are trying to develop a package that you will not be restricted by the availability of your favorite bioinformatician, and then we'll provide a robust and easy to use package so that anyone who's a beginner coder or primarily a bench scientist will be able to use our package without too much problem. So there are 4 major components that are involved when you're analyzing multiplex spatial data. So we're using a codex as well as maybe as an example here. So these are the 4 major components. The first one is the cell segmentation. You want to identify individual cells from the multiplex images. And then once you have extracted this information, you want to make sure that you want to remove some artifacts, normalize your signal, and then remove the noisy cell as well. So once you have done that, which you have a cleanup matrix that with features and cells information and their spatial location, the next step will be identifying the cell type or annotating the cell type. So in this, we definitely provide clustering that can be GPU enabled, and support vector machine, which is supervised machine learning, as well as Stella, as I just mentioned. So once you have the annotation of the cell type, the feature information, as well as the labels or the annotation of your cell type, there are many spatial analysis that are pioneered in the Nolan lab and other laboratories that enable you to extract more information and biological insights from the data. So as I mentioned, in the field, there are many packages for spatial omics that have their specific design that are very nice to use as well. But why are we different, and why are we still developing a new package, and what do we try to address? So let's start with the first one. So this is one of the things that I noticed as I was doing analysis, is that if I wanted to do an end-to-end analysis of codex image, for example, starting from cell segmentation all the way to looking at the downstream spatial analysis, I need to switch quite a few times. So in here, this table, each of the column is one steps in analyzing multiplex images. And each of the row here is a computational tool or packages that can perform parts of the steps that we need. And first one is Spacey. As you can see, Spacey is the only tool in this table that can perform from end to end without you having to switch between packages. The second thing that's also what we are proud of is that when you are switching between package, oftentimes one also need to switch between programming languages. So the popular one are some command line interface, and there's also Python, R, and also MATLAB. So as a computational biologist, maybe switching between languages is not as challenging, but as with the commercialization of the spatial omics technology, one would find that there are more bench scientists and that are interested in understanding their own data. Therefore, switching between, let's say, Python to R or from Nextflow to Python might be a challenge for a user to actually analyzing their data. So for Spacey, we will be only programming in Python, and that therefore is the barrier of entry. The last but not least, since we are working with spatial data, and there are such rich information that's within the spatial data that one can extract. So some of the downstream spatial analysis that we provide are machine learning annotations, cellular neighborhood identification, spatial context map, cellular neighborhood interface, patch proximity, and cell-cell interaction. And they are a variation of different analysis that's available. But as you can see, Spacey provides a full suite of spatial analytics that user can use readily and with relatively few steps. So the three-pointer of Spacey is first, there's no tool fragmentation. Second, it is one consistent programming language. Third, is that we have a full suite of spatial analytics. So I just want to demonstrate how Spacey is an end-to-end, multi-layered, and user-friendly analytical framework using an example as following. So the workflow goes as this. So if you just follow the arrow from here all the way down, so we start with tissue extraction. So oftentimes when people are doing spatial omics technology, they would like to maximize the space by using tissue microarray or tumor microarray. So how to extract individual ones are not that clear for a lot of packages. And the next step is on the segmentation, which I mentioned before, and data normalization is following this. And then we also have the annotation for cell type, whether through clustering or machine learning or deep learning approach. And then we also included this interactive suite for people to use to visualize their data in the way that it was acquired on top of the information that's extracted. And also this is the spatial analytics that we're providing. And most importantly, we are compatible with many other popular frameworks such as ScanPy and SquidPy. So this will allow user to switch between different packages while maintaining most of the information extracted from Spacey. So let's start with the first one. As I mentioned that in this notebook or in this demonstration, I'm using tonsil and tonsillitis as an example. So imagine this is your QP-TIFF file or your TIFF file coming out from Codex or maybe or other technology. One of the things that one might want to do is identify these individual tissues and then be able to extract them for the downstream analysis. The benefit of doing this is that you will significantly reduce the memory size that a lot of, which oftentimes pose a constraint to the downstream segmentation algorithm. And the next step is segmentation. So this is a schematic of what a tonsil tissue look like. We're basically trying to identify what are present on the slide. So to do that, we providing three different options, which are popular for the spatial omics field, which are mesmer, cellpose, and cellseg. Once the segmentation is performed and finished, within Spacey, the user can also overlay the segmentation mask on top of the original image to determine and quality control how well the segmentation is before proceeding. So once we have extracted these individual cells from the images, we often notice that for image-based technology that there are artifacts. So these can be dust or your Kim white residual, and or sometimes sticky cells. So these cells are somehow attracting a lot of antibodies and positive for almost everything, right? So this step from 8 to 11, so by the way, each step is a function. So within a few functions, you're able to do this, is to remove the artifacts and the sticky cells. So what that would look like is that imagine, well, this is the samples, the tonsil sample. We're looking at the cell size and then how many cell size, how many cells are in particular size, and removing the small cells that are basically lower than a cell's diameter or area. And the next one is normalization. So a paper that John and I, Dr. Hickey and I published earlier described different normalization, how they affect the downstream analysis. And for codecs, most of the time we'll use Z normalization, but I know for IMC, for example, ArcSine is preferred. So after normalization, we're removing the noisy cells, right? The cells are basically positive for all markers or whatever threshold of markers that the user would determine, and remove those. So now basically we get rid of the artifacts and we got rid of the sticky cells. And now we wanted to know what are the identity of these cells. And traditionally, since the feature space in the spatial proteomics is rather limited, so we do typically use clustering and using the cell type specific markers to identify each of the clusters. And within Spacey, we are able to project these, plot these cell type annotation back to their original spatial space. And then you can visualize to see whether this is befitting what you would imagine from a biological context. And then we also provide various statistics that you can use to quantify the cell type composition between different conditions for your samples. And in addition, we do also provide machine learning and deep learning approach. So this is a support vector machine training, and this is finished within, for the data set that I was using, it took less than 5 minutes. And the accuracy is pretty good on average, is above 0.8. And I'm sure with a little bit parameter tuning, you can achieve better results. And just wanted to show you in terms of what that means. So this is the annotation projected on the spatial context using SVM. And this is the same tissue and same data using cluster annotation. And you can see that more or less, I think the gross morphologies are rather similar, but definitely with the clustering, I think there's less fuzziness when it comes to support vector machine. And this also speaks to the importance when you're analyzing spatial proteomics data, that you wanted to go back to the original image to understand if this is the type of cell that you were expecting. So this is sort of going back to how important the spatial location is how important it is to the cell type identity on top of just the molecular information. And obviously we support Stella, and that is compatible with Spacey. So now we have the cell type identity figured out with various methods of you choosing. The next step that we wanted to do is providing this visualization using this visualization tool called Tissue UMAP, which is developed in a different lab. But what they are doing is really wonderful. And because codex data tend to be rather large, and using this map allows very smooth and rapid visualization and interactivity between data and images. So this is a demo where we basically open a Jupyter notebook, loading our data, the tonsil data, and loading the visualization package. So as you can see, that this is a tonsil data, you can load it in with all these different cell type annotated, right? You can click on one of them and showing this is, I would guess, the germinal center B cells, right? And then you can zoom in and then looking at this particular germinal center and then see the segmentation outline or the mask and see whether that makes sense. And then from there, you can also do various statistics and plug in, right? For example, here, you circle this area and it will automatically generate cell type proportion within the area. And there are many features that are actively being developed by the other lab, and it's a wonderful tool, and I would highly recommend for people who are working on codec and spatial data to utilize this. So once, let's say, we have visually inspected in an interactive way of the cell type identity for these spatial data, the next step, the very important part, is the spatial analysis. And one of the methods that also was developed in the Nolan lab is called a cellular neighborhood. So the cellular neighborhood, in a brief way, is basically identifying what are the tissue area or region that have very similar cell type composition that implies certain function that's shared between these neighborhoods. So more scientifically, how that is computed is that you basically, for example, in this cell paper from the Nolan lab in 2020, we have a sliding window across this tissue image by codex. And each window, you can compute the percentage of cell types that's present, right? So you basically have a cell type vector, and then you cluster these vectors based on your chosen parameters. And then the similar cell type compositions will be clustered in the same group. And therefore, based on the cell types that are enriched in that neighborhood, you can identify cell type neighborhood in this way. So using an example for the tonsil and tonsillitis data, so we have all these roughly 10, 12-ish cell type. We decided that from empirical tuning that we decided there are six cell neighborhoods that are interesting or make sense. And then we map it back to the original tissue space using a visualized color by each of individual cellular neighborhood. As you can see, so this very salmon color one here, it's enriched in germinal center B cell. And if you map it back to see the salmon color that's both in tonsil and tonsillitis, you know from biology or tonsil biology, this is the germinal center which are become more mature and fully developed during infection, which is why you see more prominent germinal centers in tonsillitis case, right? So in similar fashion, you can identify the marginal zone, and then there's B cell, dendritic cell enriched ones, epithelium, immune priming, both using the enriched cell type as well as the visualization. And this is how you identify cellular neighborhood in Spacey. There are many other ways that you could also identify cellular neighborhood, but this is the one that we supported. And just by brief look of comparing to a histology image of a tonsil, you can see that indeed, we identify germinal centers, right? And then epithelium, which is here. So I would say the robustness of cellular neighborhood identification is quite great. So let's say once we have cellular neighborhoods, how do we describe these neighborhoods' relationship in a meaningful way? So one of the methods that we propose is called spatial context. So what that looks like is almost very similar to cell type neighborhood identification, but let me walk you through it. So first, let's say if I'm looking at a window here, and let's say above certain 80% percentage, this one is mostly cellular neighborhood number 1. And similarly, let's say I'm sliding this across, and then most of this one is cell neighborhood number 2. Same is that for number 3. So this line here consists of a single cellular neighborhood. But if you go through, you will encounter places where there are multiple cellular neighborhood present, right? So this one will be a schematic where we are addressing the interface between a 2 neighborhood, and 1 and 2 here. And you can find similar mechanism or similar representation in between 2 and 3. And then obviously you will have one that's looking at the interface between the 3 of the neighborhoods. So this is basically a schematic in which that one can use to describe a particular tissue that they're working on. And we also provide the statistics on the frequency of these neighborhoods being this neighborhood, this interface being observed, so that if you have two experimental conditions, you can compare and contrast to see how are these interface or how are these neighborhood composition or schematics are different between conditions. So in the tonsil and tonsillitis case, we did exactly that. And we identified this particular combination of three neighborhoods that are significantly different in terms of frequency between the two conditions. So we expand to focus looking at these three cellular neighborhoods. So we're using a barycentric projection where each of the vertices are the neighborhoods. So what that looked like here is that for tonsillitis, we're looking at the germinal center, marginal zone, and the marginal zone for B and D cell enriched. And all of cells that are containing these three neighborhoods are projecting onto here based on where they have higher percentage towards one or the other. Right? And you can see using this barycentric projection between the two cases, you can already see that with the tonsillitis case, we definitely have a higher enrich of germinal cells and the interface between germinal center and marginal zone. So this is one way that you can explore in a much more unbiased way to explore the interfaces between different tissues, between different conditions. And then we also have a new spatial patch proximity analysis that's developed here in Spacey. So right now, I'm only showing you three neighborhood and one each. But oftentimes you have more than one neighborhood per type, right? But then the question that you want to ask is that I know there are similarity in terms of cell type composition between neighborhood one, right? But then do they share similar microenvironment surrounding these the neighborhood of the same type? If they are not similar, then what's the difference? So this is the type of question that the patch proximity analysis is trying to address. So let's say we identify the germinal center neighborhood for analysis, both for tonsil and tonsillitis. And then what we do is basically we treat these as patches, and each patch we are identifying the border cells that's outlining the patch, and then we'll compute the cells that it's in its proximity. So the distance in which that these cells will be computed is entirely decided by the user. So with that, these are the statistics one can compute to see if you draw a concentric ring around your patches, what are the cell type composition and tissue or tumor microenvironment that are different between conditions. As we can see, they are quite different and they are different between patches as well. So this is another analytical tools that one can use to infer interesting interface between patches. And there are another spatial analysis, which is what we call cell-cell interaction analysis, and mostly based on the distance. I will not go into details because of time, but basically what we do here is that we are shuffling between within the tissue, we're holding the location the same, but we're shuffling between the labels. So this way we're generating a random distribution of what the distance will be observed in random and compared to the observed one. And then you will be able to extract statistics based on the fold changes and p-value for how likely these two pairs are being observed, right? And then you can visualize that based on the condition. This is green here for tonsil, purple here for tonsillitis. So you can see the fold changes differences, and these are all significantly different ones. And you can also express this in a graph form or zooming in for the top 5 significantly different ones. So that's basically Spacey in short, quite a lot of information that's been dumped. But the key point that I want you to take home with is that it's an end-to-end Python multiplex imaging package, and we focus on tool unification. And we definitely are very robust in handling large images. These are, gigabytes, if not terabytes, large data set. And we have a comprehensive spatial analysis. And then we are really focusing on the built-in experimental setup comparison. So from there, this is basically we're looking at cell and their neighbors and how they interact together in the spatial context. The next step is our ongoing project that I'm just going to share a little bit in terms of how are these cells function together and interact with each other in the space in the 3D context. So one of the projects that's ongoing that I'm leading the effort is creating the 3D human intestine atlas. So we have a donor of different ethnicity, age, and gender, and with different coverages. We basically section 6 big intestine or large intestine and 2 small intestine, and we create a codex map and getting 30 sections, a serial section of codex. And we also do multi-omics, single-cell RNA-seq, and single-cell ATAC, and a couple with Xenium. So right now, just giving an example of the donor that I have finished processing, this is one complete donor of 29 sections. And just this one donor, we already have 24 million cells. And with 8 donors, we are looking at 200 million cells and 20 terabytes of raw data. So this is sort of the direction in which Spacey is tackling and then evolving into for the 3D component. And as I said, this is where we want to go, where we're looking at the cell type and trying to look into their spatial location, both a spatial structure, whether that be microvilli or blood vessel, and we wanted to see how they are interacting with each other and what their spatial organization. The next one is also sort of a tissue in its 3D context, but this one is slightly different. So this one is a human breast. This one is just looking at their ductal structure, right? You can see the beautiful broccoli-like branches, but this is imaged by a traditional confocal or light sheet microscopy. But what you can see, the limitation is sort of you don't really have the full comprehensive tissue microenvironment that's provided by spatial-omics. So what we do is that after we image in tissue clear 3D, we serial section it into 2D and perform codex using a 4D panel, one targeting specific for normal breast. And in this fashion, we'll be able to map the tissue microenvironment together with the continuous ductal structure to understand how the breast cancer initiation happens from DCIS to all the way to breast cancer. So with that, I just wanted to summarize in terms of the 3 projects or direction that I've talked about. So I started with the cellular identification. This is using traditional methods, looking at just molecular features or incorporating spatial information. And then within the spatial context, there are many ways. Spacey is the package that we develop to extract information from it. And the next step where we're taking this is basically looking at this from 3D perspective. And having said, presenting all these lump of information, it's not just one person work by myself. For Spacey, my talented student, Tim here, is equally contributing and on the project is super talented and hardworking and pioneer a lot of features. And we are both continuing to improve the package, both in the functionalities, user friendliness, as well as aesthetics. And then for the HubMap intestine 3D, we are working with Dr. Bei Wei and Dr. John Hickey. And for the 3D memory gland, I'm working with Dr. Hendrik Messel and Dr. Max Heist. So these are all the people that have contributed to the project that I described to you today. So with that, I would like to stop here and take questions. Thank you.
Thank you, Yuki, for the exciting presentation. I think maybe let's take a few questions, right, in the meantime. And we have, I think, actually a few questions from Kwan Nubian on the chat.
Yep.
So might I ask how the ground truth annotation is determined? That's the first question.
Yep. So the ground truth typically, I would say 99% are determined by first clustering and iteratively subclustering and annotated by like expert biologists for that annotation using marker specific annotation, right? There are also ways in which that you can do it manually by identifying which cell is which, but then given the size of the data, that's impossible. So clustering is typically the way to go. He also have a second question. Is Spacey applicable to fluorescent microscopy images such as Vectra Polaris? So yes, so I think, so I personally have not tried that, but if you are just interested, and I assume you have a DAPI, or some sort of membrane channel from that, then you can definitely use Spacey, for example, to extract tissue to do segmentation. But I think because the feature space is a little bit limited, I'm not sure how much it can get you through clustering for cell type identification and whether it's meaningful to do the downstream spatial analysis. But the short answer is yes. And happy to talk more afterwards. Did I answer your question, Xuan? Yep.
Great. Are there any questions? Sunil, I think you have one.
Yeah. So, hey, this is Sunil Badwe from Emory. I'm kind of the end user phenotype, a traditional microscopist, trying to understand how you guys do all of this. Do you think there will be ever a day where these could be plug and play kind of thing and not having to learn coding and this, that, and the other?
100%, right? I think one of the earlier vision for Spacey is that we will develop that into like almost like a desktop interface, right? Where people can just like drag and drop their images and then basically just say, I want this feature, I want this feature, which is actually available in some sort of some commercial platforms for sure, right? The difference is that this one is open source and free, and the other one is
Of course, charge your arm and a
so
leg.
yeah, exactly. But then I think it's because of the time that we didn't actually get to develop that into a desktop. But then if you actually, so we are writing this with a very detailed steps where you can basically just copy and paste. And then we have many users who's their first time coding ever, right? So I think that we have tested it to be rather user friendly. But I think there will be a day where you can just drag and drop. And then there are also ChatGPT, like a multiplex image analysis available. So we can definitely think about once Spacey is like out there, we can plug it in and then people could just use through that interface as well.
So along the same lines,
Yep.
as you were doing and showing those circular plots of
Yep.
different neighborhoods along with different germinal centers
Uh-huh.
and things like that.
Yep.
That is a really interesting way of doing things because, sorry, something else coming up on my reminder list. Okay. That's a very interesting way of doing things because there's so much heterogeneity in tissues, particularly in tumor tissues. Understanding that is rudimentary at best. So love to see you make a lot more progress in that and try to, I mean, if, I'm not trying to volunteer my services, but I think you should talk to pathologists and who might be able to ask and refine your questions for you. And there are plenty of pathologists at Stanford who would be willing and able to do that.
Yeah.
Yeah.
Sonny, thank you so much for the suggestion. That's a great suggestion. So Spacey, currently what I'm presenting here, we thinking it, publishing it or wrapping it more as a tool, but using this tool both within the lab as well as other places, we are using like focusing on biological questions such as melanoma, Merkel cell carcinoma, and then we have pathologists who are actually the driver and sample provider and the experimenter. We're working together, computational biologist and experimenter, trying to define that and what exactly are we seeing? Is that testable, both in experiment and clinic?
There was overhead announcement in the hospital, so that's the reason I was muting myself.
Ah,
Thanks, but wonderful presentation.
thank you so much.
I think we have another question just coming up. Also, Mohammed, is there any plan to develop this in R?
Well, we probably not. We actively sort of are moving away from R was because the image manipulation in terms of the support is slightly better in Python, and then the memory processing and also the deep learning tool and computer vision tools are better developed in Python. So I think that's where we'll stay. But if you're interested in converting this into R, happy to chat.
Great. Are there any other questions? So I was also wondering, I think the SPACEm is really promising for those without much data analysis expertise, right?
Mhm.
So, but there's some kind of a benchmarking that's needed. It's almost like AI, right? When you give everything to AI, what you get at the end may not be what you want. So how would you kind of incorporate these benchmarking, like human into loop steps? Like, would you still have a chat button from a bioinformatics expert saying that, am I doing it right? Right? So I'm just curious.
Yeah. I was just thinking about the other day, maybe I should start like a consulting company and just like telling people like these are the things that you should be watching out for after analyzing so many data, right? And I completely agree with you. I think human in the loop is something that it's very important for the quality control and the meaningfulness of the data, right? Ranging from cluster annotation, as well as, you know, other biological insights. So I think that's sort of a great one. I think a great project would be if you have a bioinformatician on site, and then you have who's doing the experiment and sort of discuss what is the question that we want to address and how can we use some of the computational tool listed here to develop it, right? So, but I do understand there are not many people who have access to computational biologist and whatnot. So what me and Tim, my co-developer, is trying to do is that we will have a GitHub page launch live, right? If you have any questions, maybe not about the biological interpretation, since we might not be the expert for that particular thing, but just on the coding side and whether this is the right parameters. We have detailed documentation and we'll be actively supporting, like whatever question might come related to Spacey.
Wonderful. I think there's also another question from Cohen. How robust is the cellular segmentation space, right, for tissues with very dense overlapping number of cells with lots of co-expression?
Yeah, I think, well, so since we actually didn't develop the cell segmentation method, right? So the cell segmentation is already developed and published. So there are Mesmer, CellPost, and then CellSeg. Then tonsil is the sample that we demonstrated. They're quite packed. So I would say, judging by our experience, is pretty good.
Tim, you had a question? Not. Okay. So are there any questions?
So Tim, also congratulations, because I see you are the co-author on most of this work. So congratulations, both of you have done a wonderful work.
Thank you.
So I think I'm waiting for if there's any other question. Let me see. So one more question that I have is, as we are waiting, the ChatGPT vision, right? So where are we going with this? So I see that they can generate images, but I wasn't sure that if they can analyze terabytes of data and output cell segmentation and all that. Where do you see that going?
So I don't know, like terabytes, I'm not entirely sure. But definitely I think there's one researcher that I'm following. He's also in the bioimaging sphere. His name is Robert Hess. So he actually developed like specific plugin that you can use for analysis in ChatGPT, right? But I have not tested the limit in terms of what it can take you. But there are also various analysis that people try to do using ChatGPT.
Okay, got it. And then they are not published yet, right? They are mostly discussions and initial demonstrations.
Yeah, I think for Robert's tool, it's not published. It's a Twitter feed that I saw. And for the rest, they are definitely not published. Oh, actually, there's one published from Colombia. They were using ChatGPT to do single-cell RNA-seq analysis. I think that one is published in Nature Communication, one of the journals.
Wonderful. And then regarding the data size, right? I see that when students get this terabytes of data, it's a bit of a challenge. What would be the easiest one to chop that into smaller pieces, right? Because you get that hard disk with 7 terabyte, but even one image, right? HTML file, whatever, it takes forever to load, organize, right? What would be the easiest way to handle this?
So I think the terabytes data are mostly images, right? But then oftentimes after we finish extracting them, they're sort of just kind of sitting there, either will be someday deposit into a share space for other people to use, or basically just sits on the cloud. But the image information that when extracted from these data are typically slightly less. I would say like maybe 100 gigabytes size. So that's still doable. And then what I would recommend for that, spatial data, for example, is also one of the package that we've been talking about, how to be memory efficient. So one way to basically chunk it, right, and use sparse data format. So only access the information that you would need instead of loading an entire thing into memory has been typically my approach and suggestion in doing that.
I think thank you. This is really valuable. Are there any other final burning questions? Well, with this done, let's thank to the speaker, and congratulations. I think this was a great work to you, Yuki, and Tim. And we enjoyed learning about your contribution to biological society, to especially use this. Yeah, we look forward to using this in many communities.
Thank you so much. Yeah, and if you guys have any questions, feel free to email me and Tim. I'm volunteering you here. We're happy to help you use it or discuss some of the questions you might have.
Just ask us.
Wonderful. And in the interest of time, I think we're going to wrap up early. Dr. Tan is also going to get a flight soon, right? So
Yeah.
I'm glad we made this on time. Thank you, everyone. Just before we go, final announcement is that next week we have Z Zhuang, Dr. Z Zhuang from also Stanford. I mean, we don't have particular institute selection. It's really happened to be an interesting topic. And he's going to talk about learning models for digital pathology, a complementary direction to what we discussed today. And hope to see you in 2 weeks on May 17th.
Thank you so much for organizing, and thank you all for listening.
Thank you. Thank you, Samuel. Yes, you can.